Hi, my name is Aisha Gettle. I'm an instructor in the biology department at Rio Salado College. I'm here to talk to you today about monohybrid crosses. For this example, imagine that pea plant height is controlled by one gene and that an allele for tall height, big T, is dominant to an allele for short height, little t. Imagine that a plant, which is heterozygous for height, having a genotype of big T, little t, is crossed with another pea plant that is also heterozygous for height. What are the predicted genotypes and phenotypes of the F1 generation, who are the offspring produced by this cross? This is an example of a monohybrid cross. So, how do I solve a monohybrid cross? We use Punnett squares to help us to visualize how each parent will contribute one allele per each gene of interest to each of their offspring. So these are tools which allow us to make predictions about the possible genotypes and phenotypes of offspring that can be produced by a given cross. For the provided example, we know that both parents' genotypes are big T, little t. As a first step, we would want to write out the parental genotypes, and we will write an X between the genotypes indicating this is a breeding cross. To better visualize how the alleles for this height gene are passed to offspring, we will write one parental genotype of big T, little t in yellow, and the other parental genotype of big T, little t in pink. This will help us to follow each parent's genetic contribution within our Punnett square. Note, as a general convention with heterozygotes, we always write the big letter first whenever a capital letter allele is present in any allele pair for the same gene. Now, we must remember that Mendel's law of segregation applies to gamete formation, or the formation of reproductive cells. Recall, gametes are the sperm and egg cells in humans, or the pollen and ovules in plants. So, for each parent, their two alleles for their height gene will segregate randomly, such that each gamete that they make will contain just one of their two possible alleles for the height gene. Thinking of our P height gene, where both parents' genotypes are big T, little t, for the yellow parent, 50% of their gamete cells will contain big T, and the other 50% of their gametes will contain little t. The same is true of the pink parent. 50% of their gametes will contain big T, and the other 50% of their gametes will contain little t. The Punnett square for the monohybrid cross should then be drawn. We will draw a square, then we will draw a vertical line down the center which divides the square into equal halves. Then we also draw a horizontal line across the center, which results in the larger square containing 4 equally sized internal squares. Thus, our Punnett square for a monohybrid cross looks like a 4-paned window. Next, we will write each possible allele for the pink parent over the top axis of our Punnett square. So the pink big T allele is over the first column, and the pink little t allele is over the second column. Similarly, we will write the two possible alleles from the yellow parent outside the left side axis of our square. The yellow big T will be next to the top row, and the yellow little t will be next to the bottom row of the square. To solve this Punnett square, we now must carry each individual parent's allele contribution into the predicted offspring panes within our square. We will carry each allele down through the column it aligns with for our pink parent, and carry each allele over through the row it aligns with for the yellow parent. So, for the top left pane, we will carry down one pink big T from column 1 and carry over one yellow big T from row 1 to make a pair of alleles in the pane. This offspring genotype receives one big T allele from each parent. This offspring is predicted to be homozygous dominant. For the bottom left pane, we carry down a pink big T from column 1, carry over a yellow little t from row 2. So this predicts a heterozygote. For the top right pane, we will carry down one pink little t from column 2 and carry over one yellow big T from row 1, showing this predicts another heterozygote. Finally, for the bottom right pane, we carry down a pink little t from column 2 and carry over a yellow little t from row 2. This predicts a homozygous recessive genotype. Now, we know that the possible genotypes of offspring that two parents who are both heterozygous are able to produce with respect to our example pea plant height gene. Note, this means that we also know the ratios at which each offspring genotype is predicted to be produced. To determine the predicted genotypic ratio, we need to tally how many of each distinct genotype are predicted within the square. We can see that there is one big T genotype, two big T genotypes, and one little t genotype predicted. So the predicted genotypic ratio is 1 to 2 to 1, which we can express as 1:2:1 ratio. The meaning of this ratio is that for every one offspring who will be big T, probability tells us there should be two individuals who are big T, plus one individual with the little t genotype. Expressed in another way, we could also say that for each genotype produced from this cross, there is a 25% probability that they will have a genotype of big T, a 50% probability that they will be big T, and a 25% chance that they will be little t for their pea height gene. It is also possible to express the predictions based on phenotypes or physical expressions of the alleles. In Mendelian genetics in general, recall that any capital letter allele indicates that the allele is dominant, so it will fully express itself whenever it is present in a genotype. On the other hand, any lowercase letter allele is used to indicate that the allele is recessive, which means its expression will be fully covered up and invisible in the phenotype if any dominant allele is also present. So recessive alleles will only show their physical phenotypic expression in individuals who have homozygous recessive genotypes. The implication is that homozygous dominant and heterozygous individuals will have the exact same phenotype. In the case of our pea height gene example, all offspring who have at least one big T allele will only express the tall trait and will be tall pea plants. To express the little t phenotype and be a short pea plant, a plant needs to be homozygous recessive. For our example, since big T and big T genotypes will have the exact same phenotype, we will tally them together to determine the phenotypic ratio. So, the predicted phenotypic ratio of the possible offspring is 3 who express the big T allele's effect, to each 1 who expresses the little t allele's effect. We would write this as a ratio of 3 tall to 1 short, or 3:1. We could also express this same information as a probability, or percentages, by saying there is a 75% chance that offspring will express the big T phenotype and be tall, and only a 25% chance that offspring from this cross will express a little t phenotype and be short. Questions? Do you have any questions about monohybrid crosses? If so, please reach out to your instructor for help. Learning how to effectively set up and solve a monohybrid cross looking at just one gene by using a Punnett square provides a foundation for how to approach crosses that will examine more than just one gene at a time, as in a dihybrid cross.